And one of our biggest challenges is connecting our sanctuary to land. If you notice on here, none of our boundaries actually connect to land. And so this is where programs like Stream to Sanctuary come in. Obviously, a whole bunch of different biodiversity living resources. This guy here on the bottom is the sand lance, large whales, seabirds, invertebrates, all sorts of animals. But it's also a human use area, so you'll see shipping, fishing, whale watching, recreational boating, and we're constantly trying to evolve and manage those resources and the biodiversity. And this is our research vessel, the Auk. We do all sorts of research cruises and some boater outreach for whale watching. Usually about this time of the year is when we slow down on our sea time. And yeah, here you are. So here's our annex. This is our main building, and then that's our boathouse where the Auk lives. And yeah, I don't know, Pete, if you want to just introduce yourself as well.
Just Pete DeCola, superintendent here. Looking forward to hearing about the projects. As Laura mentioned, the sanctuary is entirely within federal waters. So we struggle with connecting. Other sanctuaries are more fortunate. You can be in the sanctuary and fishing on the shoreline, and they can have their programs go all the way up to the beach. We don't have that luxury, which is why it's important to work with the whale watch industry, because they're the main avenue of access to the sanctuary. You guys are another avenue of access, so and making that important connection from stream to sanctuary. So glad you guys are here. Glad this program exists. Thanks to Jack for driving this, and looking forward to seeing your projects today. Thanks.
Yeah, I put in a few slides. I don't know if you want to add too about like our BWET grant and kind of an introduction to the Stream to Sanctuary program. This is kind of from your document that you shared. So if you want to add some words too.
Sure. Yeah. Thanks so much, Laura, and thanks, Pete, for hosting us. It's great to be back here. So my name's Jack Buckley, and I am the founder of the Cohasset Center for Student Coastal Research. So what brings us here today is the program, the Stream to Sanctuary Certificate of Watershed Stewardship. So Alex and Patrick and Michelle from North-South River Watershed Association, Scituate High School, Hingham High School, Susan from CSCR as well, Cohasset High School. There's a bunch of schools working together to engage students in community-based learning in the summertime, connecting that to school, creating some bridges between summertime community-based learning, academic year, school-time learning, and giving students, these guys who are now seniors in high school, an opportunity to put together a capstone presentation. So this work that we're doing is ongoing. It's been a grant that we've had since 2021. We have one more year in that grant, and this is something that started a little bit before our grant work began, when we connected with Ben Haskell, who's since retired. But Ben and a couple of his colleagues set the table for us, and now we're picking it up with Pete and Laura and Caitlin and everybody else here at the sanctuary. So we can talk more about that. It's, I think, a sufficient introduction for the students to share their experiences. And just as an FYI, in case the students didn't all hear that, where we have 5 minutes for each student. So if you haven't yet heard that, kind of refocus on the most important parts, because that'll take us 40 minutes, and then there'll be Q&A in which you have more time to talk a little bit more in depth.
Yeah, great. Yeah, sounds
Thanks.
Great. Amelia?
Yeah, just Amelia.
Let's see.
Laura, can you show people that QR code in case anybody wants to
Let's
also
go to the QR
access?
code. Hold on one sec. There it is, giant QR.
So all the story maps that are linked here that will be on the screen, if you also want to follow along with the story map on your cell phone, it's the link to the agenda.
Alright. school. I've done like this type of research that I'm about to explain for about 2 years now. First summer I did it with CSCR and Dr. Sarah Grady. I know some of you know her, but this past summer I wrote a grant for my project. Like I'm working with CSCR obviously, but I got $5,000 from the Margeau Foundation to buy supplies and organize like a hypothesis and recruit volunteers. So that is mostly what I'm going to be talking about today. I have a lot here. I thought originally that this was a 10-minute presentation, so I'm going to be trying to move faster, cut down. But anyway, this is my research area. I did fieldwork primarily at the Kent Street Marsh in Scituate. You drove past it today when you came down that bridge there, and you can see up there where we are. But basically, Stellwagen Bank's out there to the east, but we're mainly focusing on the marsh area here. To contextualize, my project was studying upland migration, and for anyone who doesn't know what that is, it's basically a salt marsh phenomenon, whereas sea levels rise and areas of the marsh become more covered in like salt seawater and obviously exposed to more salinity. Different grass species that can stand different salinities will end up growing inward into terrestrial areas. You can see here, parts will die off due to being drowned by the ocean and will move inland and basically migrate. And so what I'm studying for in my project is to look for a species of grass called Spartina alterniflora, which is an indicator species, and basically it grows in very salt-heavy areas. And so if that's seen out of its natural range in like high elevation marsh areas, you can tell that the marsh is migrating. And that is a sign of degradation and that the marsh probably will need some kind of conservation effort. Salt marshes obviously have a huge range of biodiversity, and that is mainly what I'm looking for in this study. I'm going to skip over mostly methodology because I know it's going to take a lot of time, but basically we used quadrats, which you can see in this photo here, which is basically a PVC pipe square, and we'd count the percentage of all grass species in that square. And then afterwards, I went and analyzed that for data. My transects that I did are here. There's 4 of them, and that was conducted based on the RASMA protocol, which basically takes the area of the marsh and divides it in order to find transects. For my conclusions on my research, I did 4 transects. And basically what I concluded was that the first transect, which was furthest up north and most exposed to washover in the main channel, had the most amount of upland migration. As you can see if you're looking at the blue here, that is alterniflora, and we're mainly looking for it to be on the edges here. But given that it's in the middle here, we can conclude that there has been formation of what's called a salt pan, which basically happens when a bit of the marsh doesn't drain properly and pools of salt water end up trapped up on the marsh, and that can lead for salt-tolerant plants to grow. That's basically a sign that the marsh is moving. The other 3 transects did not have the same level. As you can see here, mostly other grass species and not alterniflora, which I concluded meant that they were much healthier. Here's some other stuff, but I'm going to skip past that. So basically, what we're looking to protect here is the wide range of biodiversity in these salt marshes. Here's just some photos I took this summer. That's me after I fell in a ditch. Yeah. CSCR and like this 2 years of research has taught me a lot about how connected the ocean is to this town, especially as the salt marshes are huge in protecting us from flooding. As they degrade, obviously we experience more drastic effects of weather and wave action. And connections to Stellwagen Bank. I'm also the president of an environmental club at Sanctuary School, and we do cleanups a lot. And here's just a photo of some trash found in the Peggotty Beach, Kent Street Marsh area. We actually ran out of trash bags in the first hour because of how much plastic and stuff was out there, and that kind of motivated me to want to study that area and study the effects of pollution and salt marsh migration. Here's a diagram of some sources of potential pollution, such as fertilizers and oil and trash from the harbor. And skip this bit just for the sake of time. But salt marshes are crucially linked to marine environments such as Stellwagen Bank. About 75% of coastal fishery species, such as fish, crabs, crustaceans, and all that, end up using a salt marsh in some way for a source of food or as a nursery or breeding ground. And so as salt marshes degrade. As you can see up here, I have a just quote that we lose about 8,000 acres of coastal wetland every year, mostly due to sea level rise and human development. And so as we lose that, we lose crucial spaces for marine species, such as those that we want to protect in Stellwagen Bank. I wrote a final report for the Marshall Foundation. If anyone is interested in that, that is linked here, but it's like 21 pages, so I'm mostly not going to go into that now. But to conclude, basically I've learned so much these past summers, and I found like a real appreciation for what we have here in Scituate, and how privileged we are to have the protection we have at Stellwagen Bank, and how important it is we create new areas like that. Yeah, I think that's everything. I'm out of time, but thank you.
That was awesome.
Yeah.
Yeah.
I guess a couple more questions all to the end.
Yeah.
That was awesome.
Hello, I am Brian Bonagurio. I am currently a senior at Hingham High School. And since elementary school probably, I've always been the most interested in the science classes out of all the subjects at school. But until very recently, junior and sophomore year, I found that my interest above all in that field is in chemistry. And I began to look for last summer some possible opportunities to explore this passion, and I came across CCSCR through Hingham High School's summer research opportunities list. And I was interested in two projects in particular, the bacteria monitoring and water quality projects, because I believe these would be good applications of my skills in chemistry, my previous lab experience. And while this was my first year this past summer working with CCSCR, I was lucky enough to be able to work on these two projects at the same time, so I was able to total over 60 hours of time at CCSCR this summer. Areas where we conducted our research for each project are seen here, color-coded. For the watershed project, we collected samples at the locations in green along the Gulf River in Cohasset, 3 saltwater sites, and 1 small tributary that is freshwater from Hunter's Pond down at the bottom left. And for the bacteria monitoring project, well, the team additionally collected samples from around Cohasset Harbor. As one of the few people with a driver's license, I mainly did work collecting samples from 3 swimming beach locations that the Cohasset Board of Health likes to test for public safety reasons for bacteria concentrations. The bacteria monitoring project, our goals were to collect 2 samples, one for us to process at CCSCR, and additionally 1 to deliver to the Board of Health for official processing from GNL Laboratories in Quincy. Went into a bit of detail here on the procedure so that other people would be able to reference this, but I believe I'm going to have to cut it a bit short for brevity's sake. But essentially we collect 2 sterile samples from each of the Board of Health beach locations. We bring them back to CCSCR, and then using IDEX's quanti-tray procedure, we are able to get bacteria concentration estimates. A sort of brief rundown of how the procedure works is we incubate the bacteria in these trays with food that contains a molecule that is digestible by the bacteria, which then releases another molecule that
It's not you.
releases a fluorescing molecule.
There you are.
And then once that incubation period is over, we are able to see which wells fluoresce, meaning which wells are positive with bacteria, and from those well numbers we're able to get an estimate of a concentration.
Yeah, let's read it. I have explanation.
So trouble with the
Yeah.
screen. Plotted the concentrations we calculated against the concentrations calculated by GNL Labs, and we were fairly consistent with the professional laboratory. We only fell out of the 95% confidence interval of the equipment for only 5 out of the 27 times we compared samples. And this work is important to the town of Cohasset because our processing time is a few days shorter than GNL's, so we are able to return results a bit faster than GNL is and inform their decision making. And for watershed, we also collected bacteria samples there, same exact procedure with the small change that for the freshwater site we test for E. coli instead of enterococci, meaning that there is a slightly modified procedure for that, but the goal is the same. Then we additionally collect a large non-sterile sample for testing for some additional metrics of water health, including nitrates, chlorophyll A, and optical brighteners. Was really hoping I would be able to get into the procedure on this, since this is really what I find interesting. For the nitrates, we conduct this reaction using cadmium powder and solution of a few acids and molecules. First the nitrates are reduced to nitrites, and then they bind to this acid molecule, which then in turn binds to another molecule that's present in excess in the solution, forming this dye here, azo dye, named for its nitrogen double bond called an azo bond, that then produces this pink-purple color. Since these reactions are 1-to-1, the concentrations of the dye is going to be directly proportional to the concentration of nitrates, so it is then able to be measured by a fluorometer that way. Chlorophyll A, we filter set volume of sample water through a small sample using force from a vacuum pump, and then the filter is stored for processing later. And when it is it's time to process it, we dissolve the filter in an acetone solution and grind it up to release the trapped chlorophyll A particles. And then we put it in a centrifuge at a fairly low RPM to make the filter particles settle to the bottom while the chlorophyll A is still in solution at the top. Then for optical brighteners, it's a lot simpler. The sample can pretty much just be directly put into cuvettes for measuring in the fluorometer. And the final step for all of these procedures is to load the prepared samples into either plastic or glass in the case of the acetone solution, because the acetone dissolves plastic into this multi-purpose spectrometry device that we have, that using different modules is either able to either return levels or exact concentrations for each type of molecule. You can see here we're able to get exact concentrations of nitrates, but well, for optical brighteners we have to use relative concentrations. An interesting finding we had from this particular project was that at the Hunter's Pond location, where it is known that there's a lack of sewer connections in a pretty high density neighborhood, there are consistently elevated optical and nitrate levels, and this is indicative of possibly some sort of pollution there, as optical brighteners are found in laundry detergent, which can find its way into the Hunter's Pond stream there through possibly septic system leaks. And similarly, nitrates could potentially be in fecal matter that is being leaked from malfunctioning septic systems. For the importance of our ocean, I figured I should probably talk about it from like science that I understand, how I sort of look at the ocean and the problems that are faced by it. So one thing that makes the ocean important is its ability to store heat. It has a very high heat capacity. Water is a very polar molecule, lots of intermolecular forces, meaning that it takes a lot of energy to raise its average kinetic energy, which is its temperature. So for this reason, it's important for staving off drastic changes in our atmospheric temperature. It's been able to stave off climate change for a long time due to this. And for us, water temperature is an important metric to measure, because it can potentially indicate things such as higher bacteria concentration, since the optimal temperature for enterococci to survive is a bit higher than the average water temperature. Additionally, another important role of the ocean is to regulate the composition of our atmosphere. Roughly 50% of all oxygen photosynthesized on Earth comes from the oceans, which means that it is not only crucial for life here on land, but also for life in our coastal waters that we study. So you can see on this graph here, on the two of the dates where the dissolved oxygen concentrations were very high, I believe 723 and 7:9. Those dates were an incoming tide, meaning that the amount of dissolved oxygen in our waters is pretty correlated with the dissolved oxygen in the oceans. And similar to its capacity to absorb thermal energy, the ocean is capable of storing large amounts of carbon, since it is able to dissolve carbon dioxide, and which then has, when it is dissolved, a low, constant, pretty unfavorable reaction to form carbonic acid, which can then dissociate into bicarbonate and eventually carbonate. And this fully dissociated form, carbonate, is used with calcium to form calcium carbonate, and this reaction is done by many types of calcifying organisms such as mollusks and some types of plankton and coral as well. And this reaction is the most favorable under the slightly basic pH, and the historic pH of the ocean is pretty optimal for this. It allows for these creatures to survive healthily. But the dramatic excess of carbon dioxide that humans have introduced into the atmosphere disrupts this process, since the partial pressure of carbon dioxide is increased. Additional carbon dioxide dissolves into the ocean, which causes more carbonic acid to form, which, while it does increase the concentration of carbonate, it also increases the concentration of hydrogen ions by 2 times that amount, meaning that carbonate is actually less likely to form. It's more likely to turn back into bicarbonate. And obviously this is a problem for calcifying organisms, many of which live in our coastal waters. Because there's a lower concentration of carbonate, they will not be able to make their shells. As we can see on several dates here, the pH of our river is exactly correlated to their distance from the ocean. You can see on 8.6 and 7.9, they mirror their distance from the ocean perfectly, with the one closest to the head of the river being the most basic. So any effects to oceanic pH would have effects to our rivers as well.
Oh, we're just getting close on time. Oh, no.
And also, another interesting finding in one of our projects was that a potential algae bloom due to the pollution at these locations. For example, here on 7:30 we have, and 8.6 we have increased chlorophyll A levels, and on those same dates we have decreased oxygen levels, which correlate with an algae bloom. And this is just one harmful effect of pollutants. You can see here, this is just one small example of the countless streams and rivers that run into Massachusetts Bay and into the Stellwagen Bank Sanctuary. And it's important that these coastal communities have proper sewage infrastructure, and I'm glad that my work was able to bring awareness to this. I really enjoyed my time at CCSCR last summer. Obviously, a big thank you to Mr. Buckley, to all of my mentors. I was led to not only pursue the lab work that I love, but also I had to develop my skills analyzing imperfect real-world data that doesn't always match what our hypotheses are, and I think that's quite hard to replicate in a classroom environment where everything is very streamlined. And I was definitely given a new perspective on the importance of our oceans, importance of preservation, and I believe that these opportunities are like this at CSCR are vital in educating new generations of scientists or just informed citizens who are aware of the role of the ocean and understand how their watershed connects to it.
Thanks, Brian.
Hi, my name is Adri Ryan. I've been with CSCR for 2 years. A brief introduction is my name is full name is Adriana Ryan, but Adri for short. I've always been interested in the ocean and everything around it. In 2021, I got certified for scuba diving, and I've always just been intrigued by it. And CSCR gave me the opportunity to take that interest and expand it with lab work and field work and learning about the ocean near me. Just to review these pictures, this bottom right corner is us picking up trash with CSCR, top right, and this middle one we're deploying a conductivity logger in the ocean, and in this bottom left picture we are collecting bacteria samples. Our goals in the oceanography team were to educate the public on the safety hazards in Cohasset Harbor, spread awareness on the importance on keeping our harbors clean, and learning how to use scientific equipment to track and analyze the water quality, We make scientific inferences about the trends in our data, and then lastly, connect all our data work with Stellwagen Bank waters to determine these ecosystems' health. I'm gonna go over the procedure and methodology pretty quickly because a lot of it is repeated to what Brian said, with especially with bacteria. We also work with that to determine the health of the water and determine what's above the safe limit and what's below. What I think is really cool is we work with plankton as well. We have a plankton net and then usually have that underwater for 2 minutes. And then when we take it out, we look at the plankton under the microscope, as you can see here, and we either count it or identify it or both. And then lastly, our marine debris surveys followed NOAA guidelines, which can be seen here. Our purpose and community impact, again, as I mentioned, with the bacteria, it's to determine the safety of the waters, especially with recreational use. You can see here 20 of the 30 sampling dates showed healthy waters and okay for swimming, but that means that 10 had high amounts of bacteria and were not safe. We also work with pH, conductivity, and also the physical properties of water, which helps us understand how global warming and ocean acidification affect the harbor's health. A lot of our work was near local aquaculture, so our findings are highly beneficial to them as we provide reports on the health of the waters near these oyster farms. We also work with marine debris and pick up a lot of trash and record this trash. You can see we picked up 200 plus pieces on just a small strip of a beach. And you can see here like fishing gear, glass, and the majority was plastic. Lastly, with our community impact, we think we educate the community, or we do educate our community very well, especially with the State of the Harbor meeting every April. Here we present the data collected and why it is relevant to our local waters. And lastly, we hope we inspire young students in our community to take initiative on learning about their environment and being involved with their community. I'm going to go over data pretty quickly, but you can see with conductivity, the conductivity we found was between the safe ranges from 35 to 38,000. And this is salinity. If you're on this by yourself, you can like click around and look at the data, but I'm just going to go over it now. They were generally safe salinity, with the open ocean being different from the coastal waters, but that's okay. That's expected. And then here with pH and tide near aquaculture, you can see that it's generally correlated together. A healthy pH for Massachusetts waters is 8.1, and you can see it varies, but it's in a healthy range. You can see the inverse relationship between tide, or pH and temperature near the aquaculture again, and then temperature and tide near aquaculture. I'll keep going. Generally with pH, warm water equals a lower pH, and then cold water equals a higher pH. And then my favorite part, which is the plankton, you can see in this image is a cyclops copepod we found. You can see the eye in the middle, which I think looks really cool. And then here, if you slide this, these are bristleworms eating a zooplankton. I just think it looks really cool, and you can see them swimming around as we look through microscopes. Our locations, I'll just show you. We have a bunch of different locations meant for different things and different samples. If you're on this by yourself, you can like move around, and I wrote different pieces of information about each location. This is our oceanography group, and then lastly, the relevance to Stellwagen Bank National Marine Sanctuary. So through this map, you can see through the coastal watersheds go all along here, and although it's not connected by land, it's still very close to the marine sanctuary. This gray is impervious surfaces, and the more impervious surfaces there are, this increases pollution runoff, because instead of going into groundwater, it goes into the ocean. And with these high amounts of impervious surfaces near Stellwagen Bank, this can mean pollution goes further out and can impact Stellwagen Bank to a negative degree, with impacting like pH, conductivity, salinity, and all of these things, which so many marine organisms need to be in healthy parameters. Additionally, our work with plankton. So a lot of the organisms in Stellwagen Bank eat plankton as part of the food web. So our work with it also connects as with healthy amounts of plankton and healthy types of plankton near us. We can connect that to Stellwagen Bank and having an understanding if their food web is safe or not due to changes that would cause these plankton to die off. I got 50 hours over the summer, and then that's it.
Thank you, Art.
No worries. Those are cool. There you go.
Thank you. All right, hello everyone. My name is Tommy Gannon, and I've been working with Noah for a lot of my projects. So he's after me, and you guys are going to see that a lot of the stuff is kind of similar. But I'm a senior at Scituate High School, and I began my CSCR journey back in 2018. I started with the middle school program with Susan, and we've been working on two main projects over the last 6 years. The two being dune grass on Bassing Beach Island in Cohasset, and the eelgrass project in the Outer Harbor with Cohasset. But my background, I play baseball, and I'm really interested in biochemistry for college. And so where I've worked in the middle school years of CSCR, we focused a lot on Bassing Beach Island here. That's a picture of me and Noah tracking the area. But we've come back to this washover area in the middle of the island, which is where we suspect that there was like extreme storms that washed over sand and pushed back the vegetation line. But I'll talk more about that later. Another area is the former eelgrass meadows out in Cohasset Harbor. In 2022, when we started the eelgrass project, we came back from years we surveyed it every year, but the eelgrass project was ongoing long before our time. But in 2022, we came into the Outer Harbor, surveying the points, and we couldn't find any eelgrass. It was completely gone. But so these are the former eelgrass meadows. The only area that we were able to find eelgrass was sorry about that. Just present this year.
Oh, yeah,
Sorry.
yeah, just
It comes back up though.
Do we,
That's
It was working fine right before.
does it need to be his
Alright.
email, his sending?
We got it. But basically, this is Nick's Cove. This is the only area we could find eelgrass after 2022, and I suspect it's likely because of the rocks around here preventing some wave erosion from storms, and there's like deeper waters, so it's able to be insulated that way. That's where I've worked. First, I'll start off with the dune grass project. It was mostly in middle school.
So sorry. I don't know what's with this.
You can keep talking though.
But yeah,
Yeah.
basically, me and Noah, we were really just in middle school learning basic scientific skills of observation, and we're retracing the vegetation line on the beach in like the washover area. But over time, we've noticed a change recently in 2024, like this fall, a few weeks ago. Some of the middle schoolers went out and retraced the line that we had taken in 2018 and 2019, and there's like a significant change. It's not showing up on the screen.
No, what's going on here?
Basically, the vegetation line moved back farther out to where it was in the 2018 levels. It's not showing up. But basically, the vegetation has expanded farther out to the ocean, which is important because it's reinforcing the sand dunes there, which is essential for when storms come through and try to bash it. So it's been expanding outward, which has been a solid positive. The eelgrass project I started when I came into high school, and it was ongoing before my time up to 2018. But eelgrass is important because it kind of provides it's able to store carbon from the atmosphere, provide energy as a primary producer, and it provides habitat for organisms that are living out in the outer harbor. And essentially, we came in 2022 unable to find it. We decided that we would have to change our survey that we've been using to survey the population to better account for the changed environment that was out there. We decided to we added aspects of reproductive shoots, looking for indication of that, and water quality in areas with or without eelgrass, because we were looking kind of more at the future, because all of it had been wiped out. We want to see how we can potentially look at what happened and how we can help restore it in the future. But we still haven't exactly found why it's all disappeared, but we're planning on, we'd recommend that in the future. So some of the tools we use, the Biocide Pro DSS, basically tracks pH, water quality, temperature, all important things for our points in the survey map. The ArcGIS mapping software, which hasn't really been working, I'd like to show you guys some of those.
Yeah, I don't know if it's working.
Yeah, I don't know why.
It's on his laptop also. I can vouch. I saw those maps last night and they're really quite impressive.
Yeah, or if you have the QR, if you clicked the QR code earlier, if you could see it on your phone, I don't know why it's not.
Yeah, but we've also
he
had
has it though.
Yeah, we've also had the survey to track the points, mostly density of the eelgrass, and we've also used the Canway GPS trackers for the dune grass project to track those vegetation lines. Some of my sanctuary connections, eelgrass is really essential for the food web. NOAA classifies eelgrass as EFH, which is essential fish habitat, because it kind of provides small primary producing fish or primary consuming fish with area for habitat, potential foraging for food, and also as a spawning surface. And that affects the greater Stellwagen bank, as these fish are going to go out into the Stellwagen bank ecosystems and support the greater trophic levels. If there's less eelgrass, then there'll be a lower amount of fish, which will affect the amount of energy that can be sustained out in the Stellwagen bank ecosystem. And furthermore, that's going to affect people like fishermen who rely on a greater amount of fish out in Stellwagen bank to support them and their families and their communities. And that CSCR has helped me see that clearly through a greater perspective. In conclusion, at my years of CSCR, I've seen how unpredictable nature can be. You can see the vegetation might come back, it might go away. The eelgrass can just vanish in 1 year. And although it can be unpredictable, research can help us make sense of that disorder and find some more of those external factors to these changes. And there's always going to be more to learn, more to research, and I think that's important. Some research that I'd recommend for some of these future projects: continue monitoring the eelgrass and figuring out if it's coming back or not. See if there's any ways see what the best restoration method it would be for the eelgrass, and also see what may have caused it. Could it have been water quality? Is there any evidence of it still around? But yeah, thank you.
Yeah, sorry that was so funky with your images. I'm guessing maybe the computer had to be logged onto ArcGIS as well. But here's that QR code, and if you go back, you can find his presentation and look at those images. I know he worked really hard on those. If that issue keeps happening, we'll try Plan B.
Yeah, we might need to check and make sure that Noah's.
I checked signed out and it should have loaded correctly.
Yeah, but let's just see if your maps are up.
Yeah, let's look.
Okay.
That
That one worked at time, but then if it's
Down
after.
to the Shepherd. I'm a senior at Scituate High School. and I've been researching at CSCR for 7 years, same time as Tommy. I spent some time both in the middle school junior researchers project, more recently in the eelgrass/coastal ecology project. In my free time, I enjoy playing the tuba, and I'm interested in history, computer programming, and the sciences. So I'm going to talk a little bit about the projects I've been involved with. First, the Bassing Beach project kind of focused on the overwash of a 2018 storm that hit Bassing's Beach, and sand covered much of the marsh and dunes. And we've kind of tracked how the island has responded to that and how it's recovered. And then we've also tracked the eelgrass throughout the harbor, and like Tommy talked about, we dealt with the after effects of the 2022 disappearance of most of the eelgrass. Where I worked first is the Bassing's washover. Here's just a little photo of it. The AFChem is the area formerly known as Eelgrass Meadows. That's kind of a name we came up with to quickly describe it. And it used to be very plentiful eelgrass beds, but now it's effectively empty. Nick's Cove is an area in between Gull Island, Shepherd's Ledges, and the Glades. It's kind of a name we came up with. It's currently the most plentiful eelgrass beds in the harbor. And then in August 2024, we did discover a small patch of eelgrass that was growing outside of Nick's Cove in what was the AFChem. The bed is smaller and patchy, but it's kind of hopeful that there's a little bit more eelgrass. So to talk about the Bassing's project, I want to look at this image first. So that is the overwash. In a lot of sand covered up what used to be marsh there, and it was pretty void of vegetation. This image, I think, is from late 2018, early 2019. And kind of in response to this, the first thing we want to do is track the washover, so we could compare it over time. So here is a map of the washover in 2018 versus 2024. As you can see, it has a lot of vegetation has grown back, which is really positive, and it is positive for the health of the island. Greater level of vegetation means it's more resilient during storms. And it better protects Cohasset. Here's also data from Survey 123 points, which are to compare specific sites across the years. Here's data from a 2019 site and a 2024 site. Skipping ahead a little bit to the eelgrass portion, I think the first thing that when I joined the eelgrass project we were really talking about is how do we track eelgrass. It's a little more complex than I think just looking into the water, kind of for these reasons. It's really hard to see, water's often murky. The eelgrass is also patchy, and it's hard to see continuity, and it is hard to keep a boat stable amongst the various conditions of the ocean. Along with these things, we wanted to make sure any recording of eelgrass also factored in these principles, which was the inclusion of tools to make observation easier and more recordable, the ability to complete sites in a thorough but quick manner so we could complete more of them, and also versatility so that sites of different nature could be mapped equally. So in 2022, we overhauled a prior Survey 123 survey that was in use from 2018 to 2022. We streamlined it, added room for more data, and overall updated it. I'm going to skip through kind of our methodology of a site just for time's sake. To go back to eelgrass depletion, here is a map of all CSCR eelgrass sites from 2018 to 2022. As you can see, the eelgrass is pretty plentiful throughout the harbor, but post-2022, that whole area is effectively void of eelgrass, and it's only in a couple small patches. This aftermath was a big shift for the team. We kind of transitioned from mapping where the eelgrass is to mapping the conditions of it, and because there was less, so we had to, we kind of wanted to look at why that changed. So here are kind of three factors that we think could have contributed to this depletion. First is a storm. Cohasset Harbor is frequently hit by storms. We couldn't necessarily find one in the time we thought it disappeared. And we were trying to use benthic soil samples to look for root structure that would have been violently destroyed. But due to the difficulty of digging through large cobbles, we were unable to do this. Hopefully something we can try again in the future. And here's actually an image of a lot of eelgrass washed up on Bassing's Beach. We also think that this red filamentous algae that we found in 2021 and 2022 could have factored to it. A PBS NOVA documentary focusing on the Gulf of Maine did theorize that it contributes to lower water quality, and we found a lot of it in and around eelgrass. So that's another factor, and then also green crabs or other species which can destroy eelgrass. So my time at CSCR has really had me thinking about principles of ocean literacy, talking about how eelgrass is a key producer of oxygen, and also how coastal ecosystems, either through the plants or barrier islands, do a lot to protect us living in coastal towns from storms. But in the reverse, we also directly impact those ecosystems and their ability to protect us. So it really goes both ways. Kind of connecting it to Stellwagen. My time at CSR has really given me perspective into Stellwagen's mission of conserving and protecting our environment. And then this is similar to what Tommy said, but eelgrass is an essential fish habitat. So the health of eelgrass directly impacts food chains of Stellwagen. So just kind of reflecting, I think my time at CCR has given me a lot of skills that I will use throughout the rest of my life. It has helped me collaborate, think scientifically, and design experiments. I've also been really grateful to kind of recently work with some of the current middle schoolers and kind of pass on some of that knowledge. I also think generally it's taught me how coastal ecosystems are dynamic. Plants, animals, and physical structures change frequently, can be altered, destroyed, but can also rebuild. And I think it's important to understand why these changes happen and humans impacting them, especially living in a coastal town.
Nice job, Noah.
Yeah, good job. Jane, there you go.
Thank you. Hello everyone, my name is Jane Ryan and this is my CSCR experience. So, my CSCR journey, I live in North Scituate, right near the marsh. I grew up exploring the marsh, and when my sister was a freshman in high school, she joined CSCR, she would tell me about it, and I was like, that sounds awesome. So I joined the summer of 2022 after my freshman year, and throughout the summers I've kind of switched the teams I've worked with. I began working with bacteria monitoring, and then I joined the watershed quality team, and then this past summer I was a part of the ecology team, so I feel like I've really been a part of a lot of different groups, which is awesome. And these are just some photos from like the marsh. It's just so beautiful. So this is a map of where I've explored with CSCR. As I said, watershed team has taken me, as Brian pointed out, Musquashnet Brook. Hunter's Pond is right near my house. And then I kind of, as you can see, I've gone out into the marsh, explored some beaches throughout Cohasset, and then this past summer, as Tommy and Noah mentioned, Nick's Cove out here. So I've really been able to see like the stream to sanctuary directly through my work, which has been an incredible experience. So I want to start off with the bacteria monitoring team. Brian went through the whole procedure, so did Audrey, which was great. But this was really interesting, and I think one of the important parts about this group is that the data we collect is used by the Cohasset Board of Health to shut down beaches if the bacteria levels are higher than 104 mpm. So that really shows how like students have an impact on the community, and like the work we do actually has like quite a purpose in the town, in the community. So this is a graph from that summer looking at the correlation between rainfall and bacteria levels, and then cancel.
Oh, geez.
Okay. The next summer I worked with the watershed team, and here's a QR code to our formal lab report from that summer of 2023 with all of our testing that we did, if anyone wants to look at that later. But what we found is that in general, water quality concerns increase as you move up the river. So that's really because a lot of pollutants from houses in North Scituate here flow into the marsh, and then eventually that flows into the ocean, Cohasset Harbor, and then Stellwagen Bay. And this was a graph of nitrates from 2023. Nitrates can be found in a lot of sewer systems. So the green here is Hunter's Pond, which is a freshwater site, but it's in a neighborhood, so that the highest levels of nitrates were typically found at Hunter's Pond, which proves how like human impact
and potential sewage leakage is impacting water quality. So this past summer, I began my summer with an internship for State Representative Joan Moschino. And one of my first days there, she was like, what is an issue that you want to work on while you're here? And I had heard Susan in the group talking about the absolute loss of eelgrass that they had found in 2023. And I wanted to, even though I hadn't been part of the ecology team, I wanted to look into that and see the legislative side, is this a problem around Massachusetts? So for about a month or 3 weeks, I made a memo about different eelgrass policies throughout the state. The eelgrass population throughout Massachusetts has dropped to 40% of 1995 levels, so this is not just a problem in Cohasset. And during my internship, I Zoomed with the ecology team and I said, what do you guys think we can do to protect eelgrass? And they kind of explained to me it is more complicated than that, and there's not one thing we can really do. There's still so much unknown. So I joined the ecology team in July, and I kind of saw how there are so many questions that are unknown about eelgrass still. So it's very complicated, and Tommy and Noah kind of touched base on this, but we this summer was cool because we kind of found ourselves asking a lot of questions, testing different hypotheses, using trial and error. We thought maybe it could be the red filamentous algae. We tried to conduct an experiment with that. Results didn't really work out. We looked at old footage, compared that to storms, tried to see if maybe there was a big storm that washed a lot of the eelgrass population. We also looked at sediment type, which I have a map here. This is a map in 2020. A student, Lucy, made a map of a lot of the sediment type throughout Cohasset Harbor to see if there was a correlation with the eelgrass population. So we added a few points within here from this past summer, but we did not see a drastic change, so we kind of realized that's not really a conclusion either. And then, I don't know why this map here isn't loading, but we also experimented with temperature. We have the YSI Pro DSS is really cool because it has GPS tracking embedded, so we can make tracks throughout the harbor. And one trend we did notice was typically there were cooler temperatures within Nick's Cove, which is the area where the majority of eelgrass is left. So we hypothesized that maybe there could be a connection there, but there's also the rocks that kind of protect it from storms. These were some photos of the eelgrass. As I would have said, eelgrass is really essential to aquatic life and sequestering carbon from our atmosphere and ocean. So one thing I really love about CSCR is it has provided me with a hands-on education. In my sophomore year, I attended the Meaningful Watershed Education Experiences Conference with Scituate High School and Mr. Newton. And they kind of talked about how we can combine classroom and learning with outdoor hands-on experience, which I think is exactly what CSCR does. I'm taking APES this year, AP Environmental Science, and I think it's just interesting how a lot of the topics like water quality, I really get to see at CSCR, how systems are interconnected, exploring the marsh, and then the impacts of houses on water quality in the marsh, and then looking at eelgrass loss in the open ocean, and seeing how there is a connection between all of these communities, and how kind of that rippling effect on our aquatic ecosystems. So this all connects to Stellwagen Bank because if we prioritize the health of our watershed, these are all the watersheds in Massachusetts, they all flow out into the ocean and then eventually Stellwagen Bank. So CSCR has really looks into the health of our brooks, rivers, streams, marshes, but there's so many more that have an impact on our ocean as well. So this video here I thought was interesting. A few weeks ago I went to Minot Beach in Scituate, and it was absolutely covered in this red seaweed. And at one point I just like to make, before I did CSCR, I might think, ew, this is nasty, what is this? But now I feel like I look at this and I'm like, what is going on in the ocean? How is this affecting our aquatic organisms? How is this affecting levels of oxygen? And it kind of opens your eyes to the possibilities of the ocean. So my reflections on CSCR, CSCR has really ignited a passion for the environment within me. In college I hope to study environmental engineering, and it kind of, throughout my life I hope I have this responsibility to kind of protect and preserve our aquatic ecosystems, and hopefully that's what I continue doing that throughout my life. So yeah. Thanks.
job. I like how your perspective on the beach has changed. Okay, Max. All right.
I'm going to keep this extremely brief because I know we're running close on time, but this is basically going to be about my research at CSUR, which with the eelgrass team, which I'm kind of well known for. So I'm Maxwell Fernald, and I've been with CSUR for over 6 years. I initially focused on just surveying work. However, in 2022, I applied for a Margeau grant, and I won one with my team. And for that was to focus on to see if eelgrass is, or the disease that affects eelgrass called serious wasting disease is affected by salinity. And this is significant as serious wasting disease is a major infection that kills eelgrass in the 1930s, killing over 90% of eelgrass, and trying to prevent further instances of that happening. And this past year, I've been lucky enough to have 2 internship opportunities, one with the Massachusetts Division of Marine Fisheries with Forrest Shank, and the second one with MIT Sea Grant with Lily Keys, focusing on the floating wetlands projects. So for our research locations, this is basically the master map of all the eelgrass data we've taken in the past, I think, 8-ish years. As you can see, we had this large central harbor. However, a lot of this later disappeared, as we'll go on to say. This is the basement where we conducted our tank study. As you can see, it's not a very high-tech laboratory, but we did make it work, and it was a very, we needed space, and this did work, with some slight modifications. This is a lot of statistical speak, so I'm going to glaze over this a bit, but basically we did analysis of our tank study later with seeing if salinity was actually affecting the serious wasting disease amounts. This is the change in serious wasting disease per plant over 3 salinities. As you can see, we have a low positive correlation. This is
okay, it's back up. Yeah.
This is a QQ plot that just shows that the analysis we did is a good fit because we're doing linear LSRL analysis. And basically what this all means is that we had a low positive correlation with serious wasting disease with serious wasting disease being affected by salinity, which has been supported by previous literature. However, we did go to John Lefchak, who works at the University of Maryland, for further analysis, who helped us write an ANOVA script for our analysis, which is basically more complex statistical analysis. And that demonstrated that we have a fairly low confidence in our data, meaning that there could be other factors affecting our data set, but it is still in line with other literature. So I feel like CSUR has helped my ocean literacy, as it's basically taught me all about eelgrass and all about the factors that affect it. Eelgrass, as people previously said, is a keystone species that affects fish nurseries. It prevents ocean acidification. It prevents climate change through carbon sequestration and numerous other benefits. It also prevents coastal erosion. And as well, I've also learned about the connectivity of the ocean, which, as I mentioned previously, the 1930 serious wasting disease epidemic shows that the eelgrass the oceans are connected. As in that event, both sides of the Atlantic Ocean experienced 90% eelgrass loss, and that shows how literally it was all within 3 years, which shows how connected the oceans really are. For the sanctuary connections, I want to go back a bit into the eelgrass's appearance here. This is the 2017 to 2018 data for eelgrass, basically what we collected in that 2 years. And this is everything we collected in the past 3 years. So as you can see, we had this large kind of strip of eelgrass in the center, and that's pretty much all gone except for these 2 locations. And we calculate this is over a 90% area loss in eelgrass, meaning that 90% of the area that had over 75% eelgrass density have now have around 0% eelgrass density. And this is a major issue for Cohasset Harbor, as it's going to affect our water quality, it'll affect our coastal erosion. And already, at last year's State of the Harbor, a couple of lobstermen attested that this is why they're getting lower yields this year, which is a really interesting connection. Yeah. This is not an isolated incident. As previously said, 40% is all that remains of the 1990s eelgrass population of Massachusetts. As well as that, Duxbury, Plymouth, and Kingston have had a similar issue of eelgrass loss, although not to the same extent. And this has happened numerous times before in the past, and then Korea also has had a similar eelgrass loss event. And this shows that the oceans are versatile, and we do need to actually protect our ecosystems, as with climate change and all these other factors, things are rapidly dying out, like right whale populations, which does connect more to Stellwagen. So it kind of shows how the ocean is becoming more and more unhealthy, and how we have to kind of maintain its health if we don't want to have all these catastrophic effects. As a conclusion, the ocean does not seem like it's connected to us, but it very much is. A lot of things that we do are actually connected to the oceans. And eelgrass is just one example of an organism that is both necessary for our oceans, as it's provides all these benefits. It is also under distress. We also have a full documentary and full paper published under 2 different grants. So if you want to learn more about that, I'd be happy to share it with any of you. Thank you.
Oh, Matt, so is awesome. I love all your connections. Like, I never knew what an ANOVA was until college, so you're already a step ahead, so great job.
It's not like everyone's taking so long to shine, I was like, I can't take my time. Was that horrible?
Alright, you got one last, not least. Alright.
Okay.
Bring it on home, Christian.
Alright, so hey everybody, my name is Christian Bungie. I'm a senior from Cohasset High School. For my time at CSCR, I've worked both with the eelgrass and dunegrass team. Dunegrass this year, and then my junior year I helped with Max a lot with his eelgrass research into the effects of seagrass wasting disease. So that's just what I covered in the about me.
Oh my god.
Okay.
It's not
It's out of, I can just close it. It says out of memory. So if you just close these as tabs, should save you that.
And if any
I
can,
knew one of you guys would figure it
if
out.
anyone can solve problems like this, it's Christian.
Yeah, that was
Proud to be able to.
that kind of relates to my role. I served kind of like a technical role at CSCR. I worked more on like the engineering and technology side of things for both the dunegrass and eelgrass project. I come from like a background of robotics. I've served for my high school and middle school robotics team since 6th grade. That would be around 6 years now. I am currently the co-president of our high school's Rev Robotics team. So that's kind of like what I brought to the table. That's why I joined the eelgrass team. I joined CSCR in the winter of my sophomore year, though I've been involved with Cohasset Harbor for most of my life. I started sailing in the harbors out there in elementary school. I've rowed in the waters off of Cohasset since 6th grade. So it's been a fairly large part of my life. It's just my entry into CSCR has been a little bit more recent, but it's water as the harbor of Cohasset has played a fairly large role in my life regardless. So these are just the two places that I have conducted research at CSCR. On the bottom is Bassings Beach, where most of my research to do with the dunegrass team has been conducted. Then up here is just a heat map of all the eelgrass ever detected from CSCR. You can see on the bottom here, these two patches represent AFCAM, the area that was like full of eelgrass before the mass die-off observed in 2022. Then you can also see up here in this corner between rocks is Nick's Cove, the place with the majority of eelgrass following the 2022 mass die-off. So my research for the eelgrass team, these are the maps that both, I believe, Noah and Max have presented of the eelgrass pre-2022 showing widespread healthy coverage throughout the entire harbor, and then post-2022 with basically no coverage in this area and only limited eelgrass remaining up here in Nick's Cove. This is what originally motivated me to join CSCR's eelgrass team. I just found it so interesting that like in a single year, so much of eelgrass could disappear, like 90% of the density just vanished. That mass die-off of eelgrass kind of brought my attention to like the health of submarine species off the harbors I've like been involved with for most of my life, and just motivated me to join CSCR. So I was brought onto the electronics side of the eelgrass team after joining. As I said, I've had previous experience with like Arduino-based systems. I've built like an intercom at my house with my dad that used a very similar setup to what we used for the eelgrass team. So that's kind of what I brought to the project. The bulk of my time and research on the eelgrass team involved like prototyping and building a pump refrigeration system that had 4 tanks connected to relays and temperature probes that kept the water at around, I believe around like 68 degrees Fahrenheit, if I remember correctly, around that, which were about nominal ocean temperatures off the coast of Cohasset Harbor. That was just to create a controlled climate to grow eelgrass and seagrass wasting disease to test our experiment. And
Well, stuff that they're saying online, I can't see your slides.
oh,
We'll do it again. Thanks, Mary Jo.
sorry about that.
Oh, no, it wasn't. Yeah, thanks for fixing the problem.
Yeah.
That's
Yeah, sorry about that. So yeah, I was brought on for electronics. Here's a image of the proto board very early in the development. I couldn't really find any photos from later in the development. This just shows like the Arduino, I believe, I forget the exact name. It's basically the same chip as a Arduino Uno, just in a different form factor. And then the relay system we use to control the tanks can be seen right there. In addition to the pump system I created, I also worked on a computer vision model that used scans of eelgrass we collected in the laboratory to estimate percent coverage of healthy eelgrass and like dead or infected eelgrass, which we then compared to manual measurements we made just to ensure that we were getting accurate measurements on how much seagrass facing disease had spread in the lab. One of those scans can be seen here. It says 43.2% green tissue detected. It just shows the areas that it detects as healthy tissue highlighted in green. And this is one of the photos we presented at the Massachusetts Science and Engineering Fair. So me, Finn, and Stellwagen Bank intern Finn Yamini all presented our research at MCEF Region 5 and the statewide competition at Region 5. We won the Pulse of the Planet Award from NOAA, just recognizing like how our work connects to the greater environmental good, and like how we're doing is like helpful for the sustained good of the oceans. And then going into this summer, I had largely concluded my role with the eelgrass team and kind of served in a few interim positions. I initially helped out with the drifters team a bit, helping them assemble some of their project, as well as like floating some ideas for how we can possibly build some more customizable drifter platforms in the future. I also worked generally with like the CSCR file server to help make it accessible from anywhere through Google Drive's file mirroring service, just so that work would be more able to be more efficiently carried out on both dunegrass and eelgrass projects. But like after those two things, I eventually landed a role on CSCR's dunegrass team, who had been previously struggling to build a autonomous drone system to do scans of the dunegrass meadows out on Bassings Beach. Again, as I had a background in like robotics and technology, I thought that prototyping and building this drone would be a good use of my skills for this summer at CSCR. So this is just a map of the regions that we used for the drone. We split Bassings Beach into 12 roughly equal area regions that we were going to use to plot aerial scans of the drone. I display one of those paths further down, but these are just the 12 regions we use as to create those paths, and then these dots here are density points taken previously by the dunegrass team, showing like where dunegrass has been detected and at what density. And then, yeah, having previous experience with this autonomous platform called RD-Pilot, I felt fairly confident going into this project that I'd get their drone flying by the end of the summer. I successfully did that, rewiring and programming their drone to take flights in a way that was both easy to fly and could be safely conducted by anyone, even with minimal drone experience. In addition to work directly on the drone, I also helped with the development of a pipeline in Jupyter Lab that took videos recorded by the GoPro and split them up into just like screenshots at like a given interval, then geotagged them with an associated telemetry data that came directly from the drone. And then, yeah, I did that, and then those were later analyzed by Noah McDowell, another member of the dunegrass team, using a computational neural network to estimate the approximate percent coverage of dunegrass at each of these photos in the scans. This is just a heat map of all the places that our drone has flown this summer. This is generated from about 132,000 onboard telemetry pings from the dunegrass drone. This just shows the majority of places it's been. You can see some hotspots near the breakwater here where most of our missions were launched, a hotspot down to the right at Secret Beach where some of our other missions were launched from, and then a massive spot right on top of CSCR where we flew most of our calibration and test flights before moving out to Bassings Beach. The other coverage here shows the kind of like gives an indication to where we scanned dunegrass this summer. Some notable areas with like no coverage are here on the west side of Bassings Beach. Although it appears green, most of that is like tree coverage. So we just avoided scanning it to not damage the drone by like hitting a tree, and because there was no real need to scan it just because of the limited like amount of dunegrass in that area. And then here's another thing I helped design for the Dunegrass team. This was the GoPro mount that we mounted to the stand on the bottom of the drone. These two things on the side here just clipped into the drone, and then the thing on the bottom we clipped the GoPro into. And that was what we used to record all our footage this summer. This is just a quick tutorial on how we set up the flights in ArduPilot. Since this is like 6 minutes long, if anyone wants to look at it later, you can just go over it. This is just a quick video of the drone in flight. I'm going to skip over it too because we're kind of running out of time. This is a plot of one of the flights we did this summer, showing how it scanned both like regions 11 and 12. For this flight, we combined both of them just because we had a fairly full battery and felt it would be more efficient just to scan both of these regions in one flight. As you can see here, the drone keeps fairly straight paths through the air, being able to scan these areas with pretty good precision. And then these colors also just represent, the coloration of this line represents the altitude of the drone. The lighter spots represent a bit lower of an elevation, if I remember right, around 14.5 meters off the ground. And then the purple spots represent higher elevation at, I believe, 15.5 meters. The altitude for all these flights were set to 15 meters, so that shows that it stays relatively consistent to what we programmed it to do. This is a video of a flight. Do we have time? I can show like 5 seconds of it.
Okay, yeah, let's wrap. So maybe
I'll
with
show
this
like
video
5
then.
seconds of it
Yeah.
if it loads. This is just the drone taking off down at Secret Beach. I have a orange life jacket on right here. That's me. Kind of giving a command. I'm going to take the drone controller in a second. And it's going to go into autonomous mode in a little while. I think, yeah, it just went into autonomous mode there. This is basically what it did. It flew across the dunegrass meadows, just taking scans of the region that we would later analyze in the lab. As you can see here, this is all dunegrass here. Later in the video, as we pass the dunegrass, we transition into the marsh here, which, even though this isn't dunegrass, it kind of shows like how our drone was able to capture so much of the island relatively efficiently in such a short time. If you want to watch the whole video, it's like 11.5 minutes long. It'll just be on the story map after the presentation. And then for ocean literacy, CSCR, through my programs at CSCR, I've kind of expanded like my understanding of the importance of the ocean. Even though I've been involved with programs throughout Cohasset Harbor for the majority of my life, I never really considered like the greater implications of the submarine species, as well as like the species of dunegrass and other like marshland grasses that are like just off the coast that I've seen every time I've been out on the water. By doing these programs, I've kind of considered like the importance of these plants to the continued health of our harbor, and beyond that, just the health of these marine plants to the ocean in general. And then some sanctuary connections. Both dunegrass and eelgrass serve as like a bed to help prevent coastal erosion. They help anchor like the coast in place, as well as like submarine sediments, which would otherwise be swept out to sea by like currents and kind of pollute Stellwagen Bank. What's more, eelgrass acts as like a fish nursery. So by supporting the health of eelgrass in our harbor, we can help increase the density and just health of fish in Stellwagen Bank. So some other items, and here's a map of Stellwagen Bank with just the 2 local municipalities most of my research has been carried out in, Cohasset and Scituate. And just here's a map of all of Cohasset's watersheds that affect the areas of eelgrass and dunegrass here in Cohasset Harbor. So some other items I'm just going to go over quickly since we're low on time. This is a full-length documentary produced by fellow researcher Owen Gertz, going over the progress of the eelgrass team over the past few years. This is our paper as it exists right now. It's about 15 pages long if you guys have time to read that. Going over our findings on eelgrass and seagrass wasting disease. And then below this is just an Excel spreadsheet I use to calculate the drone, like some parameters for the drone flights if you're at all interested in that. And then below that is another CSV of all telemetry data pulled from the dunegrass drone. This file is rather large. It'll take probably like 2 minutes to load if you want to look at this, but I just chose to include it for completeness. In the conclusion, Throughout my work at the CSCR, I've gained like an appreciation for the vital importance of both my harbor's health, the health of Stellwagen Bank, and just like the health of the ocean in general. Even though I've been involved with the ocean for most of my life, before CSCR I've never really considered how important some of these species are to just the continued well-being of the ocean. So joining CSCR has kind of given me that context, and it's something I wish to carry with me throughout the rest of my life. Thank you.
Great job, everyone. And so, yeah, got some claps online. Yeah, we could open it up to questions. I have folks online as well. We can see you as well if you have any questions. But yeah, I'm just really impressed on all the work you did. Oh, Mary Jo, she's got the first question.
Well, first I want to thank everyone for these really engaging presentations. I've learned so much just listening to you during the short period of time, and I definitely will read all of your papers. I just wanted to say something in general because I need to leave in 2 minutes, but I'm familiar, if you looked at the photos that I think Max included, and then Christian had some photos as well, I just hope that anyone who's listening or watching that has access to lab equipment or funding for lab equipment will consider visiting CSCR and taking a look at the facilities they have, because they desperately need additional equipment. I mean, they are being super resourceful in making opportunities available to the students, but when you consider what is taking place in terms of student education and awareness and connection to the Stellwagen Bank, and then the lack of resources, I just wanted to put in a plug. If anyone has a way of helping with that, that would be great. So thank you.
Yeah, thanks, Mary Jo.
Thanks, Mary Jo.
Yeah. Yeah, any other questions? I'll pull up. Oh.
I have a question.
Oh, Laura.
This is sort of for everyone. So at our Sanctuary Advisory Council meeting the other day, we were talking with Ben Yemeni and listening to his presentation. And we started the conversation a little bit about how are we engaging more peers. And something I really didn't know about was the middle school aspect of this program, which is really great. And so I'm interested in hearing from many of you. And I feel like a lot of our outreach focuses on kind of how we can communicate with classrooms and from the adult standpoint, but how are you seeing success bringing your peers in to, you know, maybe they're not interested in joining CSCR or your research programs, but how are you getting them excited about the conservation aspect of it?
At least for my team, I found it was most helpful to not just like appeal, oh, we're doing this direct science, but say, if you were on this team, what would you want to do? For example, I got Christian by saying, we need a coder, what do you want to code with? And like, you know, or with the filmmaker, I said to my friend, hey, this might be an interesting topic to film about. So it's not about like saying, we need people to do this and this, but like, this is what your skill set is. You should join us and help us with this specific skill set.
Mm-hmm. Okay. So like they can come in and help you with pieces of it?
Yeah, they don't have to go do exactly letter of the law, you know, do this data analysis, enter into spreadsheets all day. Like if you like filming, do that. If you like doing, you know, chemistry, do that. It's really like find what you're comfortable with and go with that, 'cause it will help us, and if anything will help us.
Mm-hmm.
And I think Jane was talking about an idea of talking to other students at school.
Oh.
That's what I was trying to
Okay, I
keep.
couldn't hear you.
Yeah.
Well, we talked about like getting recognition. They just started doing like a podcast, and there's talk of this like a superintendent roundtable to kind of get the word out from the school and to the community. So I talked with the Citrus students about doing a podcast episode about like how we got involved with CSCR, kind of like our most like meaningful work that we've done and how it's kind of impacted what we want to do in our future. So I thought that could be an interesting way. But yeah, I do want to like work. I mean, I'm a senior, but this spring I hope to. I know someone from Hingham mentioned that they have like like you saw it on the research opportunities list. I feel like Scituate doesn't really have that, but I think that would be a great way to like connect students, even if they're not interested in like sciences. Like I feel like there probably are a lot of students that don't know about CSCR who would love to do it, and like this connection to Stellwagen as well. Yeah.
Oh, taking a picture.
Yeah, well, can you go over it, especially for folks that might be new to story maps, like what was the process of making the story map and what was like challenging or easy? Yeah, whoever wants to answer.
I think that's something that was kind of a challenge, but I think was also really fun, was to kind of the types of media you can include on it. Like, I think it was really interesting to incorporate the different maps, and I spent a little bit of time playing around with like different analysis tools ArcGIS has. So I think there's a lot of different ways you can represent data, and the fact that we have like a lot of data that's kind of hard to visualize, I think was just super interesting.
Yeah, just on Noah's point, like for resources that ArcGIS natively supports, like maps and like figures in there, I felt it was like sort of easy to, like easier to display it in the story map, but for some stuff in my presentation, like I present external videos I have on Google Drive as well as a paper PDF I had. I felt like that was one of the challenges I faced, was figuring out how to get the website to display those resources correctly, as they're like somewhat unconventional, and I had to kind of like shape my workflow around how can I get these things displayed in a way that I feel comfortable with and correctly communicates like what research I've done at CSCR.
Oh, great.
I don't know if it's new. I feel like I struggle to make things like horizontal. Like the story maps has a lot of like vertical, like you can add a lot of text, but I feel like it's a lot of scrolling. You can't really like format photos next to photos like what am I trying to say, horizontally, but the capability it has to like add ArcGIS, there's few platforms that you could do that on.
Yeah, I guess it's meant more to like scroll from a phone, so yeah, I see how that could be hard if you want to translate it to something bigger.
I'd say another thing about the story maps is that when ArcGIS designed it, I feel like they designed it as like a website maker kind of, less like as an oral presentation. So like when you use it as an oral presentation, you have like a lot of features that you wouldn't normally have, like because it's more like kind of like a website building software, like the maps and like being able to scroll through. It's like it's pretty interesting.
Any other questions? Great. Oh, Laura's got one.
Do you see yourself, what do you see yourself doing with this research that you have already? You know, is this your capstone, or do you find that maybe you can use this to springboard either public outreach, or, you know, I don't know if there's opportunity for signage around town and local beaches or in your schools, or bringing that into your future, your college careers? What are your next steps, if you've thought of that already?
I think CCR does a lot to help prepare for work in the field and in the laboratories. Like, we use the same sort of equipment with either like the IDEX or the YSI, so I think that's really helpful for in future studies. That's just what I found, I think.
I know he was here originally, Alex, but I also worked with him this summer to look at some other marshes, and we found that like those ones were probably more in need of restoration than when I originally studied, so I like suggest to the town that they do more focus on like separate research on that.
As mentioned in my presentation, I've been fortunate enough to work with the DMF this summer with Forrest Jank. And one thing that was really interesting is that we had our own methodology for taking data on the eelgrass plants for our study. And his methodology is almost exactly the same, even though we developed it completely independently. So it's been like really like teaching you how to find the best way to do it and apply those skills, which has been really helpful in the internship.
And then there's State of the Harbor too.
Oh, yeah.
So all students have the opportunity through CSCR to present the State of the Harbor, their research, and we invite stakeholders to that event. So hopefully that's something that lots of people will attend, and maybe we can broaden the audience for that, to get information like this out from not just our seniors, but even juniors and sophomores and freshmen.
Yeah, I was just gonna have like a more general response saying like the resourcefulness that CSCR like helps you develop. Like at least for the eelgrass project, we kind of had to work within like fairly like strong constraints with like things we already had around. Like I believe the fridge we used to help keep the tanks cool was a donation from your uncle, correct? Yeah. So we had to work with like these technologies we already had and didn't really have the opportunity to like buy specialty equipment to keep our tanks in like working order. So just having to use those things we already had on hand to create a working solution The skills like you learn from that, I feel like would be fairly useful to carry into the future in basically any job area.
I'll say like with my internship, like my time at the State House was really interesting. Like while I was there, I saw like a drug bill get passed, which like you don't realize like it's a lot of like lobbyist organizations who are like environmental. Like before my time, I kind of thought of lobbyists as like being bad, but you see how these like environmental organizations can come in with like a policy that will help the like, community, and so I saw that happen, and I kind of tried. I looked through all of like the policies that different municipalities have towards protecting eelgrass and saw how like the policies differ. So I think that like that would be a cool route for me. I think I want to go into engineering, but like the policy work behind all the environmental stuff too, I think is like a lot of interesting work.
I also think something that's really kind of unique about CSCR is how the questions we ask and the things we research is really student oriented, and it's what people are interested in. Like I think this is across kind of everyone's project. Like these are all things that like we all cared about, and we all were like, I wonder why that works, or I wonder why that happens. So I just think that's something, like the ability I've gained to ask questions and like find a way to answer them, I think will carry with me.
Oh, awesome. You got a question, Pete?
No question, just a comment. So I sit here and watch these presentations and would welcome any of you to join my staff, because your presentations are at a very, very high professional level. So I just wanted to let you all know that this is a pretty elite group here of folks, and your presentations are all outstanding. It makes me think about if there's opportunities in the future to look at marine carbon dioxide removal. Where's the chemist? There you are. Yeah. So and this eelgrass die-off, how that maybe correlates to the decline in forage fish populations that we're seeing in Stellwagen, and making those connections as well. So if I had a cat, I would take it off and tip it to you all and say, well done. And continue on, because this sort of hands-on research and your approach to getting your hands dirty is going to serve you well in college and in your career, your professional careers, wherever you choose to go. And maybe it's policy, maybe it's it's a more engineering route, or maybe it's both. You never really know. So I wouldn't close the door to anything at this point for you guys. So.
Jane's not capable of doing just one thing.
Yeah. Awesome. So, but I just wanted to thank you for letting me sit in on these presentations. It's been really, really great.
Yeah, great job. Were you connected to Stellwagen Bank at all, or knew about Stellwagen Bank before getting involved in this work? Because that's always our biggest challenge. Like, how do you actually get knowledge that our sanctuary even exists?
I think the main way I knew of it before was through whale watching, because they usually go around there, and I'd be like, why is this such a large spot for whale watching? So that's the only part of the resource where I learned of its relevance, I think.
In Scituate, we had for years and years, the middle school would run Stellwagen Day.
Yeah.
And so a lot of these seniors probably remember the now retired Miss Chambers being very excited when you were in middle school about Stellwagen Day and bringing in the salt. Is that the big inflatable whale?
Salt?
Yeah.
She's in the closet right out there.
She looks like Paula.
A lot less impressive in the closet.
I bet, yeah.
Yeah, any other questions or comments? So we do have our certificates to hand out next. Just trying to do a good way where you could also see folks on the TV. Yeah, maybe we can just like go.
Right there.
Yeah.
And the bottom.
Yeah, and actually Pete and Jack, if you want to present them. Yes.
All right.
I would be honored.
Yes. So this is our stream to Sanctuary.
Where are we standing?
I
think right in here is going to be my guess, because the camera's right there.
Yeah, I didn't think about this.
So, it's okay. You got a lot of smart kids in the room that figure it out.
Yeah, this is our Sanctuary Certificate of Watershed Stewardship. So we'll get a photo with each of you all. Here, look at all the photos.
Brian, come on up.
Wait, photo up.
We gotta do the photo.
Photo up.
Nice.
Okay, so you can come back for the team photo.
Yeah, we'll do a team photo after.
Yeah. All right,
go
Jay.
far.
Nice work. Photo up.
Photo up.
Awesome, thank you guys.
And Christian. Amelia.
Thank you.
Nice job.
Thanks. Good.
Maxwell.
Thank you.
Audrey.
Thank you.
Nicely done.
There you go. Congratulations. And Thomas. How did you? Congrats. And Noah.
Aptly named, right?
Oh, yeah.
Congrats.
Here.
Oh, yeah.
That's a good. Nice job. I just want to say thanks to Pete and everybody on the staff at Stellwagen Bank for continuing to help us with this work that we do with the students and giving us an opportunity to grow this program and learn each year how we can do it better and learn from the students. So thanks, Pete, and everybody else, and thank you for all the viewers who were here online and those who took time out of their day to join us in person, and congratulations again to all of you students for work well done.
Yeah. We need you guys to keep doing this work, because you've got to make right all the things that we've messed up in our generation. So it's your job to fix all our mistakes, so.
All right, so team
Get
photo.
cracking. All right.
Yeah.
Sure. I'll sneak in.
Can you tap? Can you step back a little bit?
Oh, yeah. All right.
There we go, good.
Was this planned on purpose to happen right before college applications are due?
Yes, I think
Yeah, that
that
is
is
the
one of
time.
the
Yes.
best things,
Oh, yes.
it's one of the best things possible for
Right at
college
the top.
applications, because
Right
it's
at the
real
top, yeah.
world, it's really who they are, and it's completely authentic. So it makes us even more grateful for this opportunity.
Yeah, thank you.
Yeah, well done, guys.
Yeah, if you have questions or any of our like,
Yeah, you got questions for us?
Yes, so I can staff questions.
Love it.
I did see a selection of Stellwagen Advisory Council earlier. I wasn't able to get in because I'm in the same town. But I'm still
So
in
they're
the
not
full
thank you so
and the meetings are, so I'd be very interested in going there.
Any member of the public can attend, you bet.
All right.
Thank you so much.
What I want to get is the Noah emblem on the podium in the photo.
Nice.
Just like
I'll send that to you in a little bit. Team Scituate. Yeah, Team Scituate photo.
Yeah, and David, if you want to say hello as well, not to put you on the spot.
We got it. Three of you here.
Laura, do you want to be in the photo?
Oh, sure.
Just getting hold of
Nice job.
Hello, everyone.
Hi there.
Hello.
Thanks so much for joining us.
My name is David Wheaton. I serve as the tribe's historic preservation officer, and I'm also a tribal councilman. I wear a couple of other hats where I
Wait, can we have your attention, please?
select board, and then I also sit on the Cape Cod Commission. I just want to say that, you know, it's really great to see young people taking an active role in trying to improve the health of our oceans and our coastal regions. I've been attending some biodiversity conferences and things, and it sounds like, you know, we have a lot of work ahead of us. And, you know, you folks, what you've done with the young people in your area to get them engaged is impressive. I'd love to learn more about how it all developed and maybe try to replicate what you've done here with the tribe. It's coast, you know, stewardship is something that's of cultural concern. We have a moral and cultural obligation to do maintain the environment for future generations to appreciate. And so I commend you for, you know, taking the initiative and having the interest. And the work that you've done looks very impressive.
Oh, thanks so much.
Thank you.
Yeah, I would love to, see if we can help get this involved with tribes and replicate it. That's kind of the goal of the project, so, yeah, we'll definitely chat more.